Creutzfeldt Jacob disease, CJD. A transmissible spongiform encephalopathy or prion disease. CJD and TSEs. Creutzfeldt Jacob disease, CJD, is one of a small group of disease classified as transmissible spongiform encephalopathies, TSEs. The leading hypothesis is that TSEs are not transmitted by microbes or viruses, but are spread by specific misshaped proteins, given the name prion proteins, CJD and TSEs. At present, only 6 human main diseases are thought to be caused by prion proteins: Creutzfeldt Jacob disease, new variant CJD, VCJD, Gertman-Strauss-Zielinski syndrome, fatal familial insomnia, Kuru, and possibly Alpers disease. CJD and TSEs. There are up to 25 prion diseases that have been identified in animals, including scrapie, sheep, bovine spongiform encephalopathy, cattle, and chronic wasting disease, deer and elk. History of CJD and prion diseases. 5th century BC. Hippocrates described disease with symptoms similar to TSE that was occurring in sheep and cattle. 4th and 5th century AD. Similar disease was recorded by the Roman Renatus. 1755. Great Britain's House of Commons discussed the epidemic of scrapie in sheep. 1759. There were unsupported claims scrapie might be contagious. History of CJD and prion diseases. Early 20th century. Despite earlier failures in attempting to show transmission of scrapie, it was finally proven through the intraocular injection of infected nervous tissue from diseased sheep to healthy sheep, which then developed the disease. First instance showing transmissibility of TSEs. 1921. The first described case of disease that became known as Creutzfeldt Jacob disease. 1950s and 60s. The disease Kuru was described and was shown to be transmitted via cannibalism. Scrapie-like lesions were found in the brains of Kuru victims. Kuru and CJD shown to be transmissible to chimpanzees. History of CJD and prion diseases. 1980s, Stanley Prusana put forth the hypothesis that a protein was responsible for the transfer of the disease. 1988, Neuropathology, bovine spongiform encephalopathy, BSE, aka mad cow disease was described in cows. Late 1980s, the prevalence of BSE in British cattle caused concerns that BSE might be transferable to humans. BSE believed to result from scrapie jumping species to cows. 1990s, a new disease similar to CJD and Kuru called new variant CJD was found in humans who had been exposed to BSE-infected cattle or their products. Case study: patient had a surgical procedure in 1971, at age 38. Determined to be the most likely source of the infection, after CJD diagnosis. Occurred before prion hypothesis or before it was believed the infectious agent was resistant to sterilization techniques. Long incubation period. The malformed prion protein induces refolding of normal proteins like itself. Slow process at first, exponential growth. Much longer replication times than microbes. It takes time to build up the levels of misfolded prion protein in neural tissue. CJD first suspected 1.5 months after first consulting physicians, 11 weeks after first symptoms. CJD diagnosis at 2 months. Diagnostic tests. Electroencephalograph. Cerebrospinal fluid analysis for 14-33 protein. MRI of the brain. Case study: final stages of disease progression, months 5 and 6. Profound dementia. Loss of speech, nearly complete inability to move. Treatment focused on comfort measures, symptom management, sedation, and prevention of opportunistic infections in hospital. Death, age 68, occurred 25 weeks after first seeking medical help for symptoms, 31 weeks after first signs and symptoms. Cause of death was pneumonia due to inability to clear lungs. Infection of others. Because of knowledge of CJD being passed by prions, appropriate precautions were taken clinically and post-mortem, so it is unlikely that others were infected after the acute stage of the disease. Transmissibility is believed to be low during incubation period. The patient was not a blood or tissue donor before or after onset of symptoms, which would have been a risk for transmission. Etiologic agent and pathophysiology. The infectious agent in Creutzfeldt Jacob disease, CJD, is a misfolded protein. This malformed protein has fewer alpha helices and more beta sheets than the normal protein. Exposure to the malformed prion protein can induce correctly folded proteins of the same type to refold into the mutant form of the protein. Etiologic agent and pathophysiology. Etiologic agent and pathophysiology. The disease state results from buildup of the malformed protein in neural tissues which kills neural cells. Incubation period can range from months to decades. The death of the nerve cells and glial cells is the cause of all signs and symptoms of CJD and other TSC diseases. Most instances of CJD are sporadic, occur in people with no known risk factors or gene mutations - unknown source. Some cases of CJD are transmitted by exposure to prion-contaminated instruments, tissues, serum, hormones, etc., from individuals with CJD, known or suspected source. The leading hypothesis. Protein-only hypothesis. The protein is believed to be the sole infectious agent. It induces its own replication by causing conformational, folding, changes in normal proteins. The two competing hypotheses. Multi-component hypotheses. Proposes prion protein is the infectious agent for the disease. Also proposes that more than just the mutated prion protein is required, cofactors also. Cofactors may include lipids and nucleic acids, but no genomic information contributes to the disease. Cofactors may form part of the prion or serve as catalysts for the refolding of normal proteins. Viral hypothesis. Postulates that an infectious virus causes the disease. Support for this hypothesis is waning. Mostly a holdover of the long-held belief that a virus must be responsible for this type of disease. Related human prion diseases. There are two inherited human prion diseases, fatal familial insomnia and Gerdman-Strauss-Zielinski syndrome. These diseases are caused by the inheritance of mutations in the PRNP gene from a parent. The mutation results in a prion protein being made which folds incorrectly without exposure to malformed proteins. There are believed to be cases of the hereditary TSCs that resulted from random point mutations in the PRNP gene, rather than being present in the genes of a parent. The human disease new variant CJD, VCJD, is caused by exposure to cows, or their products, that were infected with the prion disease bovine spongiform encephalopathy, BSE, commonly known as mad cow disease. Pathology. Human prion disease share four characteristics. 1. Spongiform changes in brain tissues. Lesions leave holes in tissue making it appear spongy. Neuronal loss, death of neuron cells. Astrocytosis, death of glial cells. Amyloid plaque formation. Some features are found in animal prion diseases. Clinical signs and symptoms of CJD. Personality changes. Psychiatric problems, depression. Lack of coordination and control. Ataxia. Involuntary jerky movements. Unusual sensations. Insomnia. Confusion. Memory problems. Final stages of the disease. Dementia. Loss of ability to speak. Loss of ability to move. Eventual death. Virulence factors. While not true virulence factors as we see in microbial diseases, there are several characteristics of all prion diseases which facilitate their transmission. Long incubation periods, months to decades. No signs or symptoms during incubation. So far, no way of detecting the mutated prion protein in living individuals. Examinations of brain tissue after death is only sure way to confirm CJD and other TSCs. Research is working on promising technology which may solve this. Virulence factors. Documented zoonotic infection of animal TSC diseases are humans. Scrapie may be the origin of BSE. BSE is the causative prion for new variant CJD. Chronic wasting disease, deer and elk, is being watched closely to see if it jumps to humans. Hunters are given advice to reduce any risk from game animals they harvest. Prion proteins are highly stable, very resistant to usual sterilization procedures. Boiling, autoclave, cooking and other heat sterilization methods don't denature the prion protein. Irradiation does not denature the prion protein. Special proteases, bleach, acid baths followed by autoclaving seem effective. We don't know how long mutated prion proteins persist on surfaces or in contaminated tissues. Some TSE may be transmissible through aerosols, tiny droplets. Modes of transmission. CJD is mostly a sporadic disease. No mode of transmission is proven to account for common form of the disease. New variant CJD, NVCJD, is believed to occur in humans as a result of exposure to bovine spongiform encephalopathy. A hereditary form of CJD, FCJD, occurs as a result of an inherited defective gene that promotes the misfolding of the prion protein with no exposure to malformed proteins. Exposure or consumption of tissues from animals with any TSE is considered a risk factor. Nervous tissue, brain and spinal cord, have highest concentration of prion proteins. Diagnostic procedures. Electroencephalograph. Cerebrospinal fluid analysis for 1433 protein. MRI of the brain. Brain biopsy. Autopsy, postmortem. Prevention measures. Feeding of rendered mammal proteins to mammals in human food chain, sheep, goats, cows, farm deer, elk, etc., has been banned in most developed countries. Brain and spinal cord are separated from carcass early in butchering process. Hunters in CWD affected areas are advised to bone out meat and avoid contact of meat with any nervous tissue. Elimination of cannibalistic funerary practices in New Guinea contributed to the end of Kuro epidemic. Treatment of CJD. No known cure, therapy or vaccine for TSE diseases. Treatment of the disease is primarily limited to treatment of symptoms and palliative care. Study of Kuro suggests there may have been resistance factors in some individuals in the population. Active research in all avenues of treatment is ongoing. Prevalence of CJD. 102 cases in Oregon, 1991 to 2009, an average of 5.7 per year. About 1 case per million people, per year, in the US of the common, sporadic, form of CJD. About 1 case per million people, per year, worldwide. Why is this an important disease to study? CJD and all other known prion diseases are fatal. Evidence suggests that prion diseases can jump species. Humans have contact with most of these species. BSE is still occasionally discovered. We eat lots of beef. Because of long incubation period, epidemics could become widespread, before signs of disease start appearing. The prion hypothesis was one of the most significant and controversial biological proposals in the modern era of biology. References. Articles, books, Belay, E. 1999. Transmissible spongiform encephalopathies in humans. Annual Review of Microbiology, 53, 238-314. Retrieved from http://www.cdc.gov/ncdod/prins/resources/belay_of_microbio.pdf. Cowan, M., and Bunn, J. 2013. Microbiology Fundamentals, A Clinical Approach. Pages 477-478. New York, McGraw-Hill. Web pages, CJD, Creutzfeldt Jacob disease, classic. ND. Retrieved from http://www.cdc.gov/ncdod/dvrd/cjd/index.htm. Creutzfeldt Jacob disease. ND. Retrieved from http://n.wikipedia.org/wiki/Creutzfeldt Jacob_disease. Oregon Health Authority. 2009, Sep 28. Oregon Rapid Deaths from Creutzfeldt Jacob disease, 1991 present. Retrieved from http://public.health.oregon.gov/diseases/diseases/documents/death.pdf. Prion. ND. Retrieved from http://n.wikipedia.org/wiki/prion. Prion diseases. ND. Retrieved from http://www.cdc.gov/ncdod/dvrd/prints/transmissible spongiform encephalopathy. ND. Retrieved from http://n.wikipediaorg/wiki/transmissible_spongiform_encephalopathy VCJD, variant Creutzfeldt Jacob disease. ND. Retrieved from http://www.cdc.gov/ncdod/dvrd/vcjd/index.htm. Photos, illustrations. Deo, S, and Deo, H, producer. 2001. CJD USA map. Print photo. Retrieved from http://www.millenniumark.net/news_files/news_news010113/news010113c.html. ND. Major regions of the human brain affected by TSI. Web graphic. Retrieved from http://wifiles.org/193%/3.html. ND. Normal to abnormal protein. Web graphic. Retrieved from http://memory.ucsf.edu/cjd/overview/biology/proteins/multiple/cause. ND. Plaques from Creutzfeldt Jacob disease in the brain. Web photo. Retrieved from http://thedallasgeek.com/2010/07/31/ Creutzfeldt Jacob disease CJD/ND. Brian theory. Web photo. Retrieved from http://lawrencecock.blogspot.com/2011/04/biology microbes proteins and prints.html. ND. Brian graphic, Mayo Foundation. Web graphic. Retrieved from http://thedallasgeek.files.wordpress.com/2010/07/r7_Korean.jpg. ND. Prints, Image 1 and Image 2. Web graphic. Retrieved from http://www.stanford.edu/group/virus/Brian/2004 ND. Deal with chronic wasting disease, CWD. Web photo. Retrieved from http://www.watch.org/news/2012/05/11500/media coverage Madcow USDA calls misleading Columbia Journalism Review calls San. ND. Sheep. Web photo. Retrieved from http://upload.wikimedia.org/wikipedia/commons/3/3d/take_hours!!!.jpg. ND. Madco 2. Web photo. Retrieved from http://www.topsecretwriters.com/2011/11/madcow disease/madco2/